The Urey-Miller experiment of 1953 provided experimental evidence for the abiotic synthesis of organic monomers under conditions that may have existed on early Earth. These monomers then polymerized to form more complex molecules, including amino acids and nucleotides. The origin of cells is thought to have occurred when these organic molecules become enclosed in lipid membranes, forming the first protocells. The evolution of prokaryotes is believed to have occurred around 3.5 billion years ago when the first bacteria-like organisms emerged. These early prokaryotes were anaerobic and used a variety of metabolic pathways to generate energy. The evolution of photosynthesis, which began around 3 billion years ago, enabled some prokaryotes to use sunlight to generate energy, producing oxygen as a byproduct. This led to the evolution of anaerobic metabolism, which is more efficient than anaerobic metabolism and allowed organisms to produce more energy. So the origin of eukaryotic cells is still a topic of debate, but it is believed to have evolved in the symbiotic merger of two or more prokaryotic cells. The symbiosis led to the development of complex structures such as nucleus, mitochondria, and chloroplasts, which are found in eukaryotic cells. The evolution of unicellular eukaryotes is characterized by the development of a variety of metabolic pathways and the evolution of diverse cell structures and functions. Many unicellular eukaryotes are capable of performing photosynthesis, while others are heterotrophic and feed on other organisms. So overall, the origin of cells and unicellular evolution is an important aspect of the history of life on Earth, and understanding this process can provide insight into the evolution of complex multicellular organisms. Next, paleontology and evolutionary history related topics. So the field of paleontology deals with the study of the fossil records to reconstruct the evolutionary history of life on Earth. The following are the major events in the evolutionary timescale. Like origin of life, the first life forms on Earth are believed to have emerged around 3.5 to 4 billion years ago. These were most likely unicellular prokaryotes. Then origin of eukaryotes. Eukaryotic cells, which have a nucleus and other outer membrane-bound organelles, emerged around 2 billion years ago. Then origin of multicellular organisms. Multicellular organisms evolved from unicellular ancestors around 1 billion years ago. So then Cambrian explosion happened. This was a period around 540 million years ago when there was a sudden diversification of life forms in the oceans, including the emergence of many of major groups of animals that exist today. Then colonization of land. Around 500 million years ago, plants and animals began to colonize. Colonization of land. Around 500 million years ago, plants and animals began to colonize land. Dinosaurs. Dinosaurs were the dominant group of land animals for about 135 million years, from around 230 to 65 million years ago. So mammals emerged around 200 million years ago, but only began to diversify and occupy many of the ecological niches left vacant by the dinosaurs after their extinction 65 million years ago. So primate evolution. Primates, which include humans, emerged around 55 million years ago. The first primates were small arboreal animals that resembled modern-day lemurs and tarsiers. So then human evolution, the genus Homo emerged around 2.5 million years ago, and our species Homo sapiens evolved around 300,000 years ago. The study of paleontology and evolutionary history is important because it provides insight into how life on Earth has evolved over time and how organisms have adapted to changing environmental conditions. It also helps us understand our own place in the natural world and our relationship to other species. So by studying the fossil record, scientists can also learn about past climate conditions and how they have influenced the evolution of life on Earth. Next topic, molecular evolution. Concepts of neutral evolution, molecular divergence, and molecular clocks and related topics. So Molecular evolution is the study of evolutionary changes at the molecular level, such as the changes in DNA and protein sequences. It has become an essential tool for understanding the evolutionary relationships between organisms and their genetic histories. Some of the major concepts in molecular evolution are neutral evolution. Neutral evolution refers to the changes in DNA or protein sequence that have no effect on an organism's fitness. These changes can accumulate over time through genetic drift and serve as a molecular clock to measure evolutionary time. So, molecular divergence. Molecular divergence refers to the accumulation of genetic differences between two populations or species over time. It is commonly used to infer the evolutionary distance between organisms and to construct phylogenetic trees. Then, molecular clocks. So, molecular clocks are based on the assumption that evolutionary changes accumulate at a constant rate over time. By comparing the differences in DNA or protein sequences between two organisms, scientists can estimate when they last shared a common ancestor. So, phylogenetic analysis. Phylogenetic analysis is a method used to construct evolutionary trees that show the relationship between organisms. Molecular data such as DNA or protein sequence can be used to infer the evolutionary relationship between species. Gene duplication and divergence. Gene duplication is a process by which a gene is duplicated to produce two or more copies of the same gene in a genome. These duplicated genes can then diverge and evolve into new genes with different functions. And molecular evolution has several important applications, including phylogenetics. Molecular evolution is used to construct evolutionary trees that show the relationship between organisms. These trees are essential for understanding the evolutionary history of life on Earth and for classifying organisms into taxonomic groups. Then, conservation biology. Molecular evolution can be used to study the genetic diversity of populations and to identify unique genetic markers that can be used for conservation purposes. Then, biotechnology. Molecular evolution is used in biotechnology to engineer new proteins with specific functions, to understand the molecular basis of diseases, and to develop new drugs. Then, forensic science. Molecular evolution is used in forensic science to identify individuals based on their DNA sequences. So, overall, molecular evolution has become an essential tool for understanding the evolutionary history of life on Earth and for addressing many important biological questions. So, the mechanisms include population genetics, populations, gene pool, gene frequency, Hardy-Weinberg law, all those concepts we will be learning. So, population genetics refers to the study of genetic variation within and among populations of the same species. The key factors that influence genetic variation within populations include mutation, natural selection, genetic drift, and gene flow. The Hardy-Weinberg law provides a theoretical model to describe how allele and genotype frequencies within a population remain constant over time under ideal conditions. So, however, in reality, populations are not static and genetic variation occur through aforementioned mechanisms. So, natural selection is a fundamental mechanism that drives evolution by allowing individuals with advantageous traits to survive and reproduce at a higher rate than those without such traits. So, over time, natural selection can result in the evolution of new species with the distinct morphological, physiological, and behavioral characteristics. Then, adaptive radiation refers to the diversification of a group of organisms into several different forms that occupy different ecological niches. Then, speciation is the process by which a single species splits into two or more distinct species. This occurs when populations of the same species become genetically isolated from each other and diverge in their genetic and phenotypic traits over time. Allopatric speciation occurs when populations become geographically separated and evolve independently from each other, while sympatric speciation occurs when populations diverge while accompanying the same geographical area. So, allopatric speciation occurs when populations become geographically separated and evolve independently from each other. But sympatric speciation occurs when populations diverge while occupying the same geographical area. They are on the same geographical area, but still they just diverge from each other. That's happening in case of sympatric speciation. So, convergent evolution refers to the evolution of similar traits in unrelated species in response to similar selection pressures. This often results in analogous structures which are similar in function but have Different evolutionary origins like sexual selection, on the other hand, refer to the differential reproductive success of individuals based on their ability to attract mates through traits such as physical appearance, behavior, and vocalizations. So, coevolution refers to the reciprocal evolutionary changes that occur between two or more species as a result of their interactions with each other. This can occur between predators and prey, parasites and hosts, or between species that engage in mutualistic relationships such as pollinators and flowering plants. So, overall, understanding the mechanisms of evolution is essential for understanding the diversity of life on Earth and how organisms adapt to changing environments over time. Then, brain, behavior, and evolution. The study of behavior is an important aspect of understanding evolution. Several approaches and methods are used to study behavior. One of the most common methods used is observation, where behavior is observed in the natural habitat of the organism. Another method used is experimentation, where the behavior of the organism is manipulated to understand the effect of environmental factors or genetic influences on the behavior. Then proximate and ultimate causation. Proximate causation is concerned with the immediate cause of behavior, while ultimate causation is concerned with the evolutionary reason or function of the behavior. And proximate causation can be explained through the study of the natural, hormonal, and physiological mechanisms underlying the behavior. Ultimate causation can be explained through the study of the evolutionary history of the behavior and its adaptive value in the organism's environment. And altruism and evolution. Altruism is a behavior that benefits others at the cost to the individual. It poses a problem for evolutionary theory as it appears to be contrary to the principle of natural selection, which favors behaviors that increase an individual's fitness. Several theories have been proposed to explain the evolution of altruistic behavior, including group selection, kin selection, and reciprocal altruism. So, group selection proposes that altruistic behavior can evolve if it benefits the group as a whole rather than the individual. However, this theory is controversial as it is difficult to explain how selection would act in the group's level. Then, kin selection proposes that altruistic behavior can evolve if it benefits close relatives who share genes with the altruistic individual. And this theory is supported by the observation that altruistic behavior is more common among close relatives. Then, reciprocal altruism proposes that altruistic behavior can evolve if individuals exchange benefits over time. This theory is supported by the observation that altruistic behavior is more common among individuals who interact frequently. So, in summary, the study of behavior and its evolution is important for understanding the adaptive value of behavior in organisms. Approaches such as observation and experimentation, as well as concepts such as proximate and ultimate causation, and theories such as group selection, kin selection, and reciprocal altruism are used to study behavior and its evolution. Then, natural basis of learning, memory, cognition, sleep, and arousal, along with biological clocks and related topics. So, neural basis of learning, memory, cognition, sleep, and arousal. The brain plays a crucial role in learning, memory, cognition, sleep, and arousal. Different areas of the brain are responsible for this process. The hippocampus is essential for memory and learning, while the prefrontal cortex is important for attention, decision making, and planning. The amygdala is involved in emotions and fear responses, and the basal ganglia play a role in motor control and learning. Then, biological clocks. Biological clocks are internal mechanisms that regulate an organism's behavior and physiology in response to environmental cues such as light and temperature. They also help an organism maintain a daily or seasonal rhythm of activity. The suprachiasmatic nucleus is seen in the hypothalamus is the main biological clock in mammals. Then, development of behavior. Behavior development is influenced by genetics and the environment. Innate behaviors such as reflexes and fixed action patterns are genetically determined, while learned behaviors are influenced by environmental factors such as experience and social interactions. Then, social communications. Social communication refers to the exchange of information between individuals of the same or different species. It includes a range of signals such as visual displays, vocalizations, and chemical signals. So, social communication is essential for many behaviors, including mating, aggression, and parental care. And social dominance. Social dominance refers to the ability of an individual to exert control over others in a special group. Social group. Dominance hierarchies are common in many animal species, including primates and birds. Dominant individuals often have access to the best resources and more successful in mating. Then, use of space and territoriality. Many animals have specific territories that they defend against intruders. Territorial behavior helps individuals control access to resources and mates. The size and shape of the territories can vary depending on the species and the environment. Mating systems and related topics. Mating systems describe the ways in which individuals of a species form pair bonds and mate. Some species have monogamous mating systems, while others have polygamous or promiscuous mating systems. Parental investment refers to the amount of time and resource that parents invest in their offspring. Reproductive success is measured by an individual's ability to produce viable offspring. Then parental care. Parental care includes a range of behaviors such as feeding, grooming, and protecting offspring. The level of parental care varies depending on the species and the environment. Then aggressive behavior. Aggressive behavior can be both innate and learned. It is often used to establish dominance or defend territory. Aggression can also be a response to perceived threats or provocation. Then habitat selection and optimality in foraging. Animals often choose habitats that provide the best resources and protection from predators. Foraging behavior is also influenced by the availability and quality of food. Optimal foraging theory suggests that animals choose the most efficient foraging strategies to minimize their energy gain. To maximize their energy gain along with minimizing the energy expenditure. So, migration and related topics. Many animals migrate over long distances to access better resources or avoid harsh conditions. Migration requires the ability to navigate and orient oneself using environmental cues such as sun, stars, or the Earth's magnetic field. So, domestication is the process by which wild animals are bred in captivity for human use. Domesticated animals often exhibit behavioral changes such as reduced aggression and increased sociability compared with their wild counterparts. These changes are thought to be a result of both genetic and environmental factors. Next, microbial fermentation and production of small and micromolecules in detail. Microbial fermentation is a process by which microorganisms break down organic compounds into simpler molecules, generating energy and producing various byproducts. This process is widely used in industrial biotechnology for the production of small and micromolecules. Fermentation is a cost-effective and sustainable way to produce various compounds such as alcohols, organic acids, enzymes, antibiotics, and many other products. So, microbial fermentation process. The microbial fermentation process involves the conversion of organic compounds into simpler molecules by microorganisms under anaerobic or aerobic conditions. The microorganisms can be bacteria, yeast, or fungi, and they utilize different metabolic pathways to generate energy and produce various byproducts. The process can be batch, fed batch, or continuous, depending on the type of product and the microorganism used. The fermentation process includes several stages such as inoculation, growth, substrate utilization by product formation, and product recovery. Examples of small and macroelement molecules produced through fermentation. Ethanol. Ethanol is one of the most widely produced small molecules through fermentation. It is produced by the fermentation of sugars such as glucose and sucrose by yeast or bacteria. Ethanol has several applications including fuel, alcoholic beverages, and solvents. Then lactic acid. Lactic acid is a small molecule produced by the fermentation of sugars by lactic acid bacteria. It has various applications in the food, pharmaceutical, and chemical industries. Then acetic acid. Acetic acid is a small molecule produced by the fermentation of ethanol by Acetobacter bacteria. It is widely used in the food and chemical industries. Then citric acid. Citric acid is a macromolecule produced by the fermentation of sugars by Aspergillus niger. It is used in the food, pharmaceutical, and chemical industries. Then antibiotics. Several antibiotics such as penicillin, erythromycin, and streptomycin are produced through microbial fermentation. These antibiotics are widely used in the medical industry to treat various bacterial infections. Advantages and disadvantages of microbial fermentations. Advantages: it's a cost-effective process. Microbial fermentation is a cost-effective way to produce various products compared to traditional chemical synthesis. Then environmental friendly microbial fermentation is a sustainable process that produces less waste and greenhouse gases compared to chemical synthesis. Versatile. Microbial fermentation can produce a wide range of products including small and macromolecules, enzymes and vaccines. So easy to scale up. Microbial fermentation can be easily scaled up from lab scale to industrial scale production. Disadvantages include contamination. Microbial fermentation can be easily contaminated by other microorganisms, leading to the production of unwanted products. It's a time consuming process. So, microbial fermentation is a time consuming process compared to chemical synthesis and specific conditions. Microbial fermentation requires specific conditions such as pH, temperature, and oxygen levels to produce the desired product. Importance of microbial fermentation in biotechnology. Microbial fermentation is an important tool in biotechnology for the production of various products. It is a sustainable and cost effective way to produce small and macromolecules, enzymes, antibiotics, and other products. Microbial fermentation has several advantages over traditional chemical synthesis, including its environmental friendliness, versatility, and ease of scaling up. Furthermore, microbial fermentation has opened up new avenues in biotechnology, such as the production of biofuels, bioplastics, and other renewable products. So, it's a revolutionizing field. Next, application of immunological principles and related topics. Immunological principles have found extensive applications in the fields of medicine, agriculture, and biotechnology. One of the major applications of immunological principles is the development of vaccines, which is used to prevent the occurrence of infectious diseases. Vaccines work by introducing a harmless form of pathogen or its antigenic components into the body, which triggers an immune response and leads to the production of antibiotics. These antibodies help in fighting off the actual pathogen when the body encounters it later. Examples of widely used vaccines include the influenza vaccine, polio vaccine, and hepatitis B vaccine. Another important application of immunological principle is in the development of diagnostic tool for the detection of infectious diseases like immunoassays such as ELISA and Western blotting are used to detect the presence of specific antigens or antibodies in biological samples. These assays have been instrumental in the detection of a wide range of diseases including HIV, hepatitis, and tuberculosis. Tissue culture is a technique used to grow cells, tissues, and organs in a controlled environment outside of the body. This technique has numerous applications in medicine, agriculture, and biotechnology. In medicine, tissue culture has been used to grow skin grafts, blood vessels, and heart valves for transplant surgeries. In agriculture, tissue culture has been used to regenerate plants from single cells, which can be used to produce disease-resistant and high-yielding crops. In biotechnology, tissue culture has been used to produce recombinant proteins and monoclonal antibodies for use in various applications. Then cell culture is a similar technique that is used to grow cells in a controlled environment. Cell culture has been used extensively in biomedical research to study the behavior of cells in vitro. This technique has helped researchers understand the molecular mechanisms underlying various diseases, including cancer and Alzheimer's disease. Cell culture is also used in the production of vaccines, recombinant proteins, and monoclonal antibodies. While these techniques have numerous advantages, there are also some disadvantages. One of the major disadvantages of tissue and cell culture is the cost associated with setting up and maintaining a sterile environment. Additionally, there is a risk of contamination, which can affect the quality of the final product. However, the benefits of these techniques outweigh the disadvantages and they continue to be widely used in various fields. So, immunology is a branch of science that deals with the study of the immune system and its response to foreign substances. The principles of immunology are widely applied in the development of vaccines and diagnostic field of various diseases. Vaccines are biological preparations that contain weakened or dead pathogens or parts of them, which stimulate the immune system to produce a protective response against the actual pathogen. Diagnostic purpose, on the other hand, involve the detection and identification of specific pathogens in clinical samples, usually by using specific antibodies. Next topic we'll be learning tomorrow. Thank you for listening.